Okay, good morning, everybody. I'd like to welcome you to the inaugural MBZUAI meeting on big model AI in drug design. It's actually the sixth AI Quorum meeting in MBZUAI from the series. I think we're going to have a great lineup of stellar speakers, a mixture from academia and industry. We have about 300 people joining us virtually on Zoom, Facebook, and on YouTube. And the way we structured this conference is going to be in pairs of talks. So we're going to have 2 pairs of 30-minute talks, and Q&A and discussion will follow after each pair of talks. And at the end of every day, we're going to have a discussion for all the speakers of that day. So I'm sure we're going to have a lovely 2 days. And without further ado, I will start and give the first lecture. My lab works on personalized medicine using large-scale human cohorts. My background is in computer science and machine learning and AI, which I've been working on for over 20 years. And I think we've seen really a revolution in the field of medicine coming from, driven by data. And the first example of this is in the year 2000, when we had the publication of the human genome. We went, if you think about the 20-something years that have passed since then, we've actually went from knowing the involvement of a few genetic variants for some diseases to now, 20-something years later, we know the involvement of hundreds of genetic variants, sometimes even thousands of genetic variants for every disease. And not only that, but when genetic evidence is used in the development of drugs, it's been shown that that actually helps in reducing significantly the cost of drug development by an order of about 50%. But genetics is, of course, only just one piece of the puzzle. It's what we're born with. It doesn't change with life. It doesn't take into account environmental factors, physical activity, nutrition, everything else that goes on in life. And that's why I think that the next leap in understanding human health and disease is going to come from modeling additional or measuring additional data, mainly taking into account all these other environmental factors. And for that reason, a few years ago, we started what we now call, like we had the Human Genome Project, we call this now the Human Phenotype Project, where the idea is to go now very, very deep and measure everything that we can, that we know how to measure today about individuals. And I believe that such an approach is going to allow us to identify molecular signatures of disease before the disease appears. So it'll allow us to predict disease ahead of time, to identify trajectories that people are following on for disease. In some cases, it will also allow us to find the drivers of disease, so to identify therapeutic markers. And this data will take us to hopefully personalized, AI-based, and even remote healthcare. So what is it that we actually measure? So every participant that joins the study comes to a clinic that we set up at the Weizmann Institute and undergoes this battery of tests. On the right-hand side, you can see all the clinical and physiological tests that we do. And on the left-hand side, we have the molecular tests and the biobanking. So I'll walk you through this very briefly. So before people come to the clinic, they upload all of their historical medical records. So we have their historical blood tests. We have all of their past diagnoses. Then on site, we do additional blood tests. We use ultrasound to look at fat in the liver and to look at the carotids for cardiovascular health. We use a DEXA machine to look at full body composition and bone density, EKG and ABI for cardiovascular health. We take high-resolution images of the retina, from which you can see blood vessels, and that's also considered to be a window to the heart. We use continuous glucose monitors that for 2 weeks measure every 15 minutes glucose levels in the blood of people. And then we use sleeping devices, which are at the grade of a sleeping lab that tracks analog signals throughout the night, from which we can identify various measures of sleep quality and I believe that has never been studied on a group of thousands of individuals, definitely not coupled with all of this other information. We then know all the medication that people are taking. They log their diet, so we have their dietary intake. They fill out hundreds of questionnaires on medical background, lifestyle, physical activity, and we have all their past, current, and future diagnoses because we track this cohort longitudinally. Then on the left-hand side, you can see all the molecular tests that we do. So we do full whole genome genetic sequencing for each participant in the study. We profiled the microbiome, both the oral and the gut microbiome. We use mass spec to look at thousands of different metabolites in the blood, what's called metabolomics. We use from the blood also, we're looking at protein assays to look at proteomics, as well as on RNA sequencing from blood cells. And we have a novel immunology assay that we adapted from the lab of Steve Elledge that allows us to synthesize hundreds of thousands of antigens. And then with phage display, we can take antibodies from the serum of a person and identify all of the antigens that are bound by a person in a single experiment. And this we also multiplex and do at high throughput for the cohort. And then we biobank. We have about 160,000 samples right now that we biobanked from, including PBMC, serum, and plasma. So all of this we've profiled and measured so far on, actually it's over 10,000 people, approaching 11,000 people in Israel. This cohort, the baseline cohort, is a cohort of healthy individuals between the age of 40 to 70. But in addition to that, we have about 2,000 individuals from various diseases that we've profiled in collaboration with clinicians in Israel, various oncology cohorts, a diabetes cohort, obesity, cardiovascular health, and several others. And we follow this cohort longitudinally. Every 2 years, participants come back to the clinic and get all this profile done again, except, of course, for the genetic profiling. So I'll show you in a moment some of the discoveries and insights that we've been identifying from this data, but because we believe that there's a lot of value in this data, we're looking now to actually expand this significantly on several different dimensions. So one dimension is to get to the next milestone of increasing the number of people, to go from 10,000 to profiling 100,000 people, to look at additional diseases, additional ethnic groups, and so additional geographies, additional cultures. And although we are doing very deep phenotyping of individuals, there's also, of course, additional tests that we are not doing, so to also increase the phenotypes that we measure. So this is the vision going ahead. And I believe that such a data set is going to have tremendous interest and impact because I draw on the very impressive and very impactful project of the UK Biobank. So this is a cohort that started about 15 years ago. 500,000 people tracked longitudinally in the UK. But this cohort is tracked mostly for electronic health records, genetics, and very little multi-omics. So it's much more shallower compared to the depth of measurements that we do. Nevertheless, if you look over the years at the impact that the UK Biobank has had, it's really been incredible. So there's over 8,000 publications that use the UK Biobank that were built on top of the UK Biobank. About 30,000 researchers from 100 countries, major investments by companies to increase the size of the data set. So it's really had tremendous impact. Just last year, over 2,000 publications published using the UK Biobank. And our data, even though it's smaller at the moment, it's still growing, as I mentioned, and it will likely be the first cohort that will be international. So such a profiling will be done in multiple countries. And it includes all these other tests shown here, microbiome, RNA sequencing, the immune profiling, metabolomics, and so on, that are not part of the UK Biobank. So I think that the depth of this data set is going to bring a lot of interest. And in the coming months, we're actually going to open this data set for research, both to researchers in academia as well as to industry. And this is going to be on a secure cloud-based environment where you cannot download the data, but you can research it in whichever way you want. The data is, of course, anonymized. So what I'd like to do now is tell you a little bit from really a bird's eye view, some of the insights that we've been gaining from this data. Obviously, for lack of time, I won't be able to go into all of this, so it'll be just to touch upon some of the various different projects. And the first one I'd like to start with is a focus that we have on the microbiome, where we're trying to identify bacterial strains that we believe will have therapeutic value. And so the idea is to develop a pipeline that includes a novel computational approach for discovering bacteria that may have therapeutic benefits. I'll mention the computational pipeline in a moment. But once we identify the relevant bacterial strains, we are working to then go and isolate them from those individuals in the cohort that we believe harbor the beneficial strains, and then formulate them into a rationalized probiotics, if you will, and then go into clinical trials to see if they actually have efficacy for the indication we developed them for. And the first two that we're developing are for type 2 diabetes and weight loss. And the idea is that because of the size of the cohort, which is the largest microbiome data set coupled with all of these other metadata information that we have, it allows us to go beyond just looking at the composition of which bacteria are there, but really to dive deeper into single nucleotide variation in the bacteria and associate that with various traits. So the idea is much like we're doing in genome-wide association studies of human genetics, where we're looking for SNPs in the human genome that correlates with traits. Now we want to look at SNPs in bacterial genomes that correlate with traits. So there's a lot of nuances and adaptations that have to be done to the pipeline from human genetics, but roughly the idea is that we take metagenomic samples, so deep sequencing of stool samples, we map them onto bacterial reference genomes, but then we record the variation that we see at each individual position. So for example, the variation highlighted in blue there, it's just an illustration, of course, but for these three individuals, you can see that the individual at the top has an A at the highlighted position and a BMI of 31, you can see on the right, whereas the two other individuals have a T at that position and BMI, a lower BMI. So this, for example, would be an association between variation at that position and the trait being BMI in this case. Of course, this is an illustration on 3 people. We do this on the 10,000 person cohort that we have, and then we get these Manhattan plots that you're used to seeing from human genetics, except that now the x-axis is not human chromosomes, but these are bacterial species. And every position here on the x-axis represents a position within a specific bacterial genome, where variation at that position had an association to BMI in this case, where the p-value is shown on the y-axis. So what's striking here is that the associations are very significant, so we find many significant hits between variations at even just single nucleotides at individual bacterial species with the trait. But I think what's more interesting than that is actually not the significance, but the effect size. So the number of BMI points that are statistically explained by this variation, which you can see here in showing the same data but as a volcano plot. And you can see many individual SNPs that explain 1 or 2 points of BMI. That's a huge effect. It can be 5 or 10 kilograms of body weight. And when we look at individual strains, we can see, for example, here in the middle, this is one bacterial species where we identified 2 strains. For simplicity, let's call them the blue and the green strain. And individuals harboring the blue versus the green strain have, on average, a difference of 2 points of BMI, which is a huge effect. And not only that, but when we look at what happens 2 years later, we see that those individuals that harbored the blue strain associated with lower weight did not have a change in weight, whereas individuals harboring the green strain associated with higher weight, BMI gained on average another point of BMI. So the idea is now to isolate these blue strains And we know the individuals that harbor them and take additional strains like the one here that also have similar effects, package them into a consortia of bacteria, and go into clinical trials in humans where we'd like to see if they have efficacy. We've done a similar trial, not with kind of precision probiotics where we isolate strains, but with a more crude approach of doing a fecal microbiome transplant, so transferring the entire bacteria from healthy donors to diseased patients. And this we did in a disease called atopic dermatitis, a disease of the skin, very prevalent in about 20% of children, about 10% of adults. We published this last year. It was a proof of principle study on only 9 patients. We're now in the midst of expanding this to a larger set of patients. And you can see here an individual which after week 4, after given placebo capsules, you can still see clear manifestations of the disease on this person. And after providing the FMT, we see clinical clearance of the symptoms. And this is for another patient where even weeks later, many weeks after we give the last dose of the FMT, we still see clearance of clinical symptoms. When our clinicians scored this clinically, it's called a SCORAD, score of atopic dermatitis. On the x-axis, you can see that after week 4, where each person being its own control received the placebo capsules, there's no change in the score, and the score drops then on average by 70%. It actually improved for all 9 patients after the first 2 doses of FMT. When we analyzed the microbiome, we could see when we reduce it by a t-SNE plot to these 2 dimensions, we can see that the samples of the donors, highlighted here in ellipses, are far away from the samples of the patients initially, before we actually do the FMT. After we do the placebo, there's really no change in the microbiome of the patients, as there shouldn't be. But once we begin to actually do the FMT, we see that the patients have a major shift in their microbiome, and their microbiome becomes not identical, but much more similar now to their donors, and this happened for the vast majority of patients. So we believe that it's this FMT intervention which actually resulted in the clinical benefits, and in this case of atopic dermatitis, we're now trying in the larger study to chase after those particular bacteria that we think gave the therapeutic beneficial effect, and then also isolate them and see if such a formulation may serve as a treatment for atopic dermatitis. I'll briefly mention another area of the microbiome that we're looking at, and this is the effect of medication that effect that medication have on the microbiome. So as we know, medication that we take have many side effects, and they're mediated by multiple factors, and we wanted to investigate whether one of them could be actually effects that the medication have on the gut bacteria. So here too, our cohort was very instrumental, because we have over 2,700 people out of the cohort that are taking some type of medication. So we documented these, and then we looked at linear regression models that incorporate various covariates like sex, age, BMI, and so on, and still saw an effect of medication on the abundance of some gut bacteria, like some highlighted here. For example, people who take metformin have an effect on this Shigella species, but there's many such associations. And because we know that gut bacteria play an instrumental role in our health and disease, then we believe that medication that we take that have an effect, a direct effect on the presence or abundance of some bacteria, they may also exert side effects or have additional impact beyond the impact that they have on the original target for which they were developed. Another area that we're looking at is the blood metabolomics data that we're measuring. So this is with mass spectrometry. We can identify thousands of different molecules that we look at in blood. And so a couple of years ago, using this cohort, we published work where we tried to identify what are the determinants of each of the different metabolites that we measure. And the advantage of having a cohort where we have very diverse profiles of many, many different types of data is that we can compare the relative contribution of genetics, microbiome, diet, lifestyle factors, and so on, we can compare the effect of each of them on each metabolite. So what we did was to develop machine learning models, either linear models or gradient boosting decision tree models for each metabolite. And what we found is that each metabolite was about over 90% of the metabolites could be significantly explained, and typically it was one of these factors that provided a much better statistical explanation compared to the others. So for example, in the metabolites shown here, you can see that the four on the left are very well explained in held out samples by composition of gut bacteria, whereas metabolites in the center here are best explained by diet. A simple positive example here is levels of caffeine in the blood are mostly explained by how much coffee people drink, so an expected example, but of course many other metabolites have much less known associations. And then some metabolites, like the one on the right, are mostly explained by genetics. And so the reason why this knowledge map is key and important is because we know that many metabolites that circulate in our bloodstream are key for disease and health. And so if we identify some of these metabolites and we'd like to modulate them, then these would be the factors that we would need to likely change in order to modulate these metabolites. So for example, metabolites here on the left, if these were key for some disease, they're probably going to be modulated by changes to the gut bacteria. But then equipped with this knowledge map, we can go and shed light on specific diseases, on pathophysiology of some diseases. And this is an example from a paper that we published last year on acute coronary syndrome patients who underwent a heart attack. And the idea is that you can use this knowledge map to gain insight even if you have just a single patient. And the idea is that we have our acute coronary syndrome patient, and let's say it's a 60-year-old male. We can go into our healthy cohort and identify 60-year-old males who did not undergo a heart attack. So these would be matched healthy controls, specific, personalized for this patient. We can then go and look in the metabolomics data, and we can build a reference for what the metabolite level should be for each metabolite in this personalized reference cohort for this patient. And then we can see where the level of this metabolite falls for our patient. And in this case, for example, it falls quite far off from the expected distribution of 60-year-old males. And of course, we can do this for all of the different metabolites that we have. And then we end up with a metabolite deviation table for all of the metabolites in this patient. And now that we have this table, we can cross it with the knowledge map of metabolomic data that I mentioned before and see if the metabolites that deviate in this patient, are they mainly metabolites that are driven by diet, microbiome, genetics, lifestyle factors, or and so on. And when we do that, we can see that even though we have two patients, say two 60-year-old males who present with the same clinical endpoint of a heart attack at the hospital, when we analyze their metabolite deviations and cross it with the knowledge map, we can see that the underlying factors are likely different, and therefore the treatment that we should give patients should be different. So in some people, it may be that it's the diet that may have caused the metabolite deviations and perhaps the disease, and in others, it could be genetics, in other, it could be the microbiome, and so on. Okay, another example that we've been using this data is to study aging, specifically biological aging. So here, I'll start with an example of a model that we built from the metabolomics data. So what we did was we took the metabolomics data and we built a machine learning model for predicting the chronological age of people. And this is the result of this model and held out data. And you can see that the model using only metabolites data could do a fairly good job at predicting the chronological age of a person. But of course, when you look at this, the predictions are not perfect. And at every age group, we can see individuals where the model predicted an older age than their actual chronological age, or a younger age than their actual chronological age. And so we asked whether these predictions are just artifacts and errors in the model, or whether perhaps when the model sees that a certain individual has a higher, or the model thinks it has a higher age, what it means is that their metabolites actually look like those of an older person, and maybe that's indicative of faster metabolomic age, higher metabolomic age, higher biological age in that person, and maybe that is indicative of worse clinical condition. And indeed, when we look at the people highlighted in red, which have, according to the model, a higher predicted metabolomic age compared to people in green who have a younger metabolomic age, we see that individuals with this accelerated aging are indeed in worse health condition comparing parameters that were never taken into account in these models, including they have higher BMI, higher lipids, so higher triglycerides, higher hemoglobin A1C, blood pressure, higher cholesterol, and higher liver enzymes. So we think that these metabolites here that we know which ones are driving the predictions in each person. Maybe these are markers of accelerated aging from the metabolomics data. But we have a very deep profile of individuals, and metabolomics is just one of the different body systems that we measured. So we took all of the data and we categorized it into 14 different body systems that we measure. Each body system is measured sometimes by multiple different types of measurements and features. And we then tried to build these models of chronological age from each of these different 14 body systems. And the results on the top show that we could actually build these biological clocks, if you will, from each of these different body systems. So each gave a significant prediction, significant ability to predict chronological age. Interestingly, we could predict chronological age better in females compared to males. And we also looked at the bottom here, the correlation between these different clocks, and we found an interesting result that surprised us, which is, by and large, these clocks are independent, so there's very little correlation between them. It means that if your body is aging faster in one system of the body, it doesn't necessarily mean that you have faster aging in another system of the body. Moreover, we saw that these clocks have likely clinical meaning, because when somebody has a higher biological age for a certain body system, that individual is typically at higher risk for developing a disease, but not only a general disease, but a disease related to the body system on which the clock was developed. For example, if we look at the clock for insulin resistance, we see that individuals who have a faster aging of their insulin resistance clock, they would be at a higher likelihood of developing diabetes, which is related to this clock, and so on. So these clocks are, again, largely independent. They also likely have clinical meaning. We also found, I believe for the first time, a difference between males and females. In males, we see a kind of a linear decline of, in this case, it's the metabolomic age with chronological age. And in females, you can see that although we have this decline, there's this abrupt change around the age of 50, around the age of menopause. And we actually saw that menopause likely induces certain significant dramatic changes on some body systems, because when we take even a matched cohort of women at the same age, but those who passed or those who didn't pass yet menopause, we can see that certain body systems, certain profiles of body systems, the biological age, could separate quite well menopause versus not. We also see that by and large, sex differences between males and females on these body systems, by and large, they diminish with chronological age. So the ability to take these clinical omic profiles and separate between males and females, that ability somewhat degrades over time. So males and females become more similar to each other. And we identify some metabolites that have interesting patterns of change with age. Sometimes some metabolites don't change with age, some do, some are similar between males and females, and some differ between them. Lastly, I'll mention just very briefly that using the continuous glucose monitoring data, this is work that already we published several years ago, we showed that people have very different, very personalized responses to meals, even when they eat exactly the same meal. And then using clinical information and microbiome data, we were able to develop an algorithm that predicts these changes, and then to use this algorithm to prescribe personalized diets for people that take individuals which normally on their habitual diet have these huge changes in spikes in glucose levels, and then with the same amount of calories, we could prescribe diets for them that balance their blood sugar levels. And we recently performed randomized clinical trials on people with prediabetes and diabetes, showing that if you follow these diets for half a year, you improve clinical endpoints much more than the standard of care approaches. So with that, I just want to end with seeing the insights that we're learning from this type of data. We believe that such a data set can really have tremendous impact. A lot of discoveries could be made from it. So we are looking for many different ways to now expand this. I mentioned before, we'd like to increase now the number of people, and the next milestone is to do 100,000 people, which we hope to achieve by building additional cohorts, expanding the cohort in Israel, but also there's several additional countries, including here in the Emirates, in which we are looking to expand and build these cohorts. We'd like to expand to more diseases as well as to additional phenotypes. And we developed everything in the lab, so full stack from really the operations of how do you recruit a cohort, how do you build it, the clinic in which people go and get profiled, then the high throughput automated robotics to do the multi-omics at very high throughput and low cost, including some proprietary assays. And then, of course, the core of my group is on the data science and data analysis. And we are looking with this platform to collaborate and expand to doing measurements in additional countries, additional organizations. And overall, I really believe that this type of data is what is going to take us to the next generation of precision medicine. And with that, I will end and just put the most important slide of all the people in my group who are really doing tremendous work: students, postdoc, research coordinators, lab technicians, driving this research. Thank you very much. And so, as I mentioned, now we won't have Q&A, and we'll go straight to the next talk from Shai Shenor from the Technion.
Okay. Just, should I press? Good morning, everyone, and thank you. I'd like to thank the organizers for the invitation. So what I'm going to be talking about today, and I think really kind of closely, it was a good pair to talk with Iran, is something I call the data insight gap, right? And how do we close it? Just to kind of, from an affiliation perspective, my name is Shai Shenor. I'm a Faculty of Medicine member at the Technion. I'm also the Chief Scientist of Cytoreason, and I head the biomedical arm of the Tech AI, the umbrella organization of the Technion for AI. And this point of the data insight gap is something that has been troubling me for many years. I've been doing computational biology, systems biology for well over 20 years now, almost a dinosaur, as some of us in the room. and really, it's a half glass full, half glass empty piece, because when we started doing this, the human genome wasn't yet sequenced. Iran showed you a slide from early 2000 to start off with genotypes. And that even wasn't a reality, and we kept continuously looking at the excitement of what the revolution of genomics is bringing us. But the truth is that there's a half-glass empty side to this as well, right? And that's the fact that data has been basically exploding exponentially. The ability to measure new stuff has been coming and becoming something that we embrace new technologies to do this all the time. But data isn't the ultimate thing we seek. What we seek is insight, or the value of what that data is insight. And that isn't growing at the same scale, right? So I call this the data insight gap, and I think for us who've been doing kind of computational systems biology for a while, I say the problem with this is really that if you look at this, is that the gap means that you can't scale it in the same way we've done it to date, right? The central dogma of bioinformatics is basically one data set, one PhD student generating one paper, right? But that basically means that the percent of data that gets actually kind of utilized drops. And this is a problem that exists in every industry, but the more complex the thing we're studying, and biology is very complex, the bigger this gap, right? And so one needs to think about how do you actually change the relationship here between data and insight. And to do this, I'm just going to touch on a few of the things that make this gap, right? Why aren't we really getting the bang for the buck from the data? And so 3 points here, and I'll just show you some examples of work that we've done on this. One is what I would call technology mismatch, right? The fact that we measure the data not necessarily in the same way that we actually want to be thinking about the biology. The second is the knowledge avalanche, namely that we've been exploding in the number of papers we put out as well. And last is that we keep doing things from scratch. We learn, we basically do this, we start from ground zero again versus actually building on everything that we've done to date. So here's an example of the technology mismatch. This is one that I think everybody would easily get. And that is really that cells are the atomic unit of biology, and yet there are many, many different measurements that we don't get at the cell level. In fact, I would argue that it is such a fundamental concept and such a technology mismatch is that the reason that we like single-cell data isn't necessarily that it's really high-quality data. It's because that's the atomic unit we want to be thinking in, right? And we are willing to tolerate noise and sparseness of the data and so forth just to be able to think in this space. I'll show you an application of this. So here's a paper we published a few years ago, taking 3 publicly available data sets from IBD, inflammatory bowel disease, 30 Crohn's patients and ulcerative colitis, 30 of which, all of which got anti-TNF, infliximab, and 30 of which, half of which responded to the treatment and half did not, okay? Very standard for many different drugs. These are data sets that have been around since 2009 in the public domain. When we did this work, there were 5 papers published trying to predict who's going to respond to anti-TNF or not. Those 5 papers found different gene signatures, none of which were overlapping, amazingly enough. And what you can do if you take this data set, so these are colon biopsies from baseline pre-treatment of patients measured on Affymetrix array, right? Old technology. And if you just look here, oh, this is nice. You can see a volcano plot of all the genes, differentially expressed genes at baseline between responders and non-responders. This is a meta-analysis of the 3 data sets, right? Now, you take this, there's hundreds or thousands of genes differentially expressed. You're trying to make sense of what does it mean to be, kind of a responder versus non-responder to kind of a biomarker for anti-TNF. You take the same data and you deconvolve it. So you reconstruct the cellular composition of every patient, and you can generate a volcano plot that basically where every point is no longer a gene, it's a cell type that you estimated from the data of the gene expression. And what you can see easily is that responders and non-responders really have a big difference at baseline between 2 different cell subsets, plasma cells and macrophages. This is a much more easily interpretable understanding, an insightful understanding of this, right? And the way I normally say it is that gene expression, at least not at the single cell level, is an expensive, confusing way to count cells, right? Not only can we say what are the cell subsets that differ at baseline, we can also say how the regulatory programs of these cells differ. And this particular insight, we were able to, for the first time, after these data sets lying around for 10 years in the public domain, validated independent cohort from Ichilov and Rambam patients, where you can basically see staining for plasma cells, and you can, in fact, by plasma cell staining, differentiate responders and non-responders with an AUC of 82% when you control for inflammation. So that's an example of a technology mismatch. It's a very simple one we can all get, and it's one of many, right? So here's another example, more recent, thinking about time. We collect cohorts, we collect patients in a discrete way. You know, you come in on day 2, you come in on day 14. There's a lot of clinical reasons why we do this, right? Logistical reasons why we do this. But in fact, biology is continuous, and we want to be thinking about this in a continuous manner. And when we don't, we actually lose a lot of information. So what we've done is developed algorithms that allow us to basically reconstruct the continuous nature of disease progression, of temporal changes in disease. This is a paper that's currently under review. It's one of 3 that we've recently published on this, all of which base the ideas of sequence alignment, right? Old school stuff. Now take this to the idea of disease trajectories, right? So trajectories, and how can you align trajectories to one another? We call this algorithm Timex. You basically take patients, each of which have collected at different time points, each of which have a different profile, and the disease is heterogeneous. It changes between patients at different rates. And what we can now do is, rather than consider them based on the discrete time points in which they were collected, rather we assemble the disease progression trajectory from the data. We can do this on a group level. We can do this for subpopulations and so forth. And what Timex, the algorithm that does this, actually does is multiple trajectory alignment. So we basically construct a consensus trajectory. Every patient has a few time points. A few time points here, and we can now construct the trajectory of how the disease changes over time and predict per person where they sit on the trajectory, and account for this as another way of stratifying patients. So basically, the algorithm takes in longitudinal data from multiple individuals, originally 3 samples, but now we can drop this to less, identifies a seed of genes that change over time, but we can do this for any data, including microbiome, and find those kind of behaviors that look similarly in patients despite the heterogeneity. This is the seed of genes that kind of change, so there are phase shifts and so forth, random versus random, and use this to basically construct a multiple trajectory alignment. And once we've done that, we basically have a consensus trajectory. We can project people and validate with independent cohorts. This is the idea. Just giving you an example here, a small publicly available data set of bladder cancer patients, 18 patients, 84 samples over 15 years. Okay, 15 years of bladder cancer data. You take this original data, you try to analyze, say, tumor purity, simple measurements that you can deconvolve from gene expression data. This is RNA-seq data from the bladder, and you can see there's basically very high kind of heterogeneity here, no clear signal. You build the disease pseudotime, you suddenly have a continuous trajectory with an inflection point of pre and post. This information you can now use to position people on the trajectory. Taking this to TCGA, you can now predict outcomes of bladder cancer better than what you can do with staging. So this is the type of data. We can do this for short vaccination studies. We can do this for large things such as cancer over 15 years. In any type of data, this is possible to do. So rather than thinking about the chronological component, we use the data to build the disease progression component, and then we can ask, how does this explain information? So just differing this from some of the stuff that Eran showed. Okay, so that's just one gap, right? Technology mismatch for cells, for time, just makes getting insight from data more difficult. Here's another one, thinking about knowledge. You can think about, having, papers coming out. When I was a grad student, when I was a master's student, I read every paper that had the word network in it. Every paper that was published in PubMed. Now, my field is immunology. Every 30 minutes, a new paper gets published. Every 30 minutes, right? Now, if you think of half our crap, you're still not doing well, right? You're just not going to be rereading everything. You somehow need to move from this to the right? And what we've done is move this to an NLP system. This was done a few years ago. We're basically thinking about the literature at a cell level. Here are the cells. this is what they're doing. If we're thinking about a protein-protein interaction, this is the cell that it occurs in. This is what the cell responds to. This is what the cell secretes. Everything in the literature needs to fit how we think about biology and put this in a way that's standardized and can be connected with the data. Just showing you this as one slide. We're taking this across all of PubMed. 182 diseases shown here on the left. Every point is one disease. This disease is basically described by the cells that has been associated with it, the cytokines that those cells secrete, and so forth, all of which have been identified in the context of that particular disease. And we can compare them and identify similarities of diseases based on the immune-centric view of the literature. Given this, for instance, we have a clustering of a bunch of diseases here that fall together in this module 10. And what you can see here on the right-hand side is, so all of these diseases, for instance, have an association with CCL8 as a chemokine, except for psoriasis. We validated CCL8 is, in fact, in gene expression data associated with psoriasis. This has never been picked up in the literature. So you can, again, use machine learning to basically bridge this gap on the literature side. Last, I said another gap is learning from building on data that you've already done. And we've taken this as an example in my lab to think about the big problem of the cross-species gap. Right? How do you overcome the fact that we sometimes have to do experiments in animal models and we want to be curing humans? Right? And every time we do the mouse experiment, on the experimental side, we've been investing a huge amount in humanizing mice. What have we done on the computational side? Close to nothing. So what we've done is basically develop an algorithm we call FIT. And what FIT does is, rather than take the mouse data and just try and understand how it relates to human, it takes all the publicly available data that we've collected to date on human disease models, or human diseases and mouse models of those diseases, learns what the relationships looks like in those previous experiments done by giants, and then, given a new mouse model, extrapolates to what we'd expect to see in the human. Okay? So just kind of showing you, this is a very, the first version of this we published in Nature Methods, 2018, looking, for instance, in any one gene. You can build a database of what the effect size would be in human, disease versus control. What is the effect size in the mouse model of this disease? We pair human diseases and mice models, and you can bootstrap this relationship to learn the relationship at every gene level between how a gene behaves in the mouse and how you'd expect it to behave to human. Given this prior information, now we can take a new mouse and look at its gene expression data, a mouse we've never seen before, a mouse experiment we've never seen before, and estimate what we'd expect to see in the human. This is what you see here with the distribution of this, so that we can ultimately give a prediction of what we're expecting in human and what's the confidence interval of this. Just doing this very simple thing, it was our first attempt to do this, we can increase cross-species translation between 20 to 50% of the overlap. So a mouse gene that if you see a mouse differential expression, we can move to telling you if it's going to be, if you just take that and directly translate it to human, we can do basically 50% better than what you would do this way just by learning from previous data. You can see this in any multiple different fold changes here on the bottom versus the true positive fraction of predicting the overlap between mouse and human. And what we can basically see is that in any confidence interval, we actually do better using the prediction of the fit model than we would have just using the mouse data. And here's an experiment just showing you an example of a gene that no mouse model actually said is highly differentially expressed. No human disease data show that it was differentially expressed, but the machine learning model learned from all the mice experiment that when this gene is slightly upregulated in the mouse, it's going to be highly upregulated in the human. We bought a protein for an antibody for the soul of a gene I have never played with before, for the sole purpose of checking this in IBD, and you can see here again that indeed ILF3 is differentially expressed in IBD as predicted by the model, and not observed in the data directly in the human before. So just gave you 3 examples of this idea of the data insight gap. But there's really hundreds of these ideas, right? Hundreds of ways in which we can bring algorithms to bear that increase the insight from the data. And the problem is that if you think about this, we've been doing computational biology for 25 years, give or take, right? At this point, we basically have an algorithm for each one of the different kind of data types that are out there. We've been building newer and newer versions of this, and there's really nothing there's nothing new under the sun. The problem really is how do you actually integrate all those individual great algorithms and papers that many of my colleagues here have published into one integrated framework that will bring utility, that would change the central dogma of bioinformatics, so that no longer would we be starting and having this equation of a single data set and a single person doing a paper, right? Because that doesn't scale. And so what the concept I thought of was basically this idea of how do you build an integration framework that I call computational disease models. It needs to have 3 arms. One is to bring in all the big data that's out there, right? Put it in the natural form that we think about the data, so there's no technology mismatch. The other side is to bring the knowledge, all the literature. It needs to talk to one another, as I showed you before. And the third arm is the machine learning component that needs to iterate and improve this every time a new paper comes out and every time a new data set comes out. And this is the idea that we aim to do. And the reason we thought this would work is because it's worked in other industries, in other areas that are way simpler than biology. Think what happened in, say, the navigation industry over the past 25 years, right? We basically moved from maps. My children don't even know what this thing on the left is, right? To something that was semi-useful, right? And then we took a smartphone and we put a model of the real world on it, and it basically transformed our lives. We can no longer drive without this taking the phone, and in fact, if we forget it at home, we go and we run and pick it up again, right? And we just now layer more and more data on this. In the same 25 years, biology basically moved from being an analog field to having big data, but that big data is still in a way that isn't really useful. And so what we need to do is to build this computational disease model. And to do this, I started Cytoreason back in 2016. So Cytoreason today is a pharma AI company. It's a tech company geared at building computational disease models to enhance drug development. It's currently about 130 people and has some of the kind of blue chip industry that we've been working with for several years now and growing over time. And the idea is basically how can we change the data insight gap to accelerate the ability to actually make drug development faster and more accurate. And there's obviously a business side to this, right, if you think about this. And the business side of this is that the return on investment on R&D in pharma is basically negative, right? So it costs us way too much to develop drugs. And if you think about how and this is a barrier, a clear barrier for precision medicine, because if I don't have a blockbuster and you want me to develop for a small subpopulation, I can't fail, right? And the reason this is so negative is because most drugs fail. So what we need to do is somehow use the insight from data to de-risk drug development. And this is basically what Cytoreason does across the board, is build a system that allows us to make drug development more accurate by modeling these, kind of putting together, pre-packaging all the data in a given disease and all the literature in a very useful new data type, which is these computational disease models. So for instance, right now we're modeling the cell composition per disease, the cell composition of every patient, the cell state, the intercellular tissue network, how the disease changes over time, what is the clinical variation of individuals within that and subpopulations and so forth, and how do treatments affect those individuals. Imagine for every disease having this pre-packaged even before you started developing the mouse model that you're looking for, right? This becomes something that's highly useful to actually perform drug development. And so with this idea, we went to kind of look at the industry and realize there's another problem, and that is that this industry has basically been built with silos of data. Every drug company has its own data set, right? Very few people have, and actually no one has what Ron was showing us, right? This multi-omic large cohorts is really difficult in particular diseases. Even if you have a large data set, you have it in one indication where you had a successful drug and you know nothing about the other drugs, other diseases that you potentially may need to go into the market. And this is a nice quote I picked up from the AI lead in J&J, where he basically said, we believe we have the most data out there, and it's 3% of the data, right? Now think about how Google makes an ad placement with about 50% of web traffic, or Amazon offers you a book with 60% of web traffic, you're trying to basically cure disease with no data. And so what we've developed on this framework of computational disease models is simultaneously a system that allows us to learn summary statistics information from every data set that we see, so that the disease model grows with the number of samples that are actually building this kind of correlation structure of what the disease looks like. And you can see this here for one indication, and we build this per disease and subpopulation. And what you can do with this now is something we call synthetic trials, right? So what a synthetic trial is, imagine that you have the compilation of all the disease, all the data in a particular disease for multiple different diseases. How would you use it? Well, so there's basically two options to use it. One option to do so is what we call an umbrella trial, right? Which is where people take multiple different assets that they have and try and compare them to see which one would work best in a particular disease, right? The other one is to actually pick which disease and disease subpopulation would be relevant for you, and that's called a basket trial in the industry. So basically, give me I have an asset, and let's now rank all the indications which we want to be going after for clinical trials. Now, the nice thing is, because everything here is in silico, you can do this with data or without data, right? So you can do this, say, indication prioritization for a target that you haven't even generated data for, or you can do this as an indication expansion once you've had a successful clinical trial and you want to choose what the next indication is. And so you can basically take the entire understanding what you have in a disease with you everywhere, just like we take the phone to every decision-making that I need to make during the very expensive course of developing new assets. And so this is what we've been using the system for, and I'll just show you an example. This is a publicly released example of our first collaboration with Pfizer back in 2017. Pfizer came to us and says, look, we have a CCR6 inhibitor. CCR6 is a chemokine receptor. We want to figure out, we have no human data on it. We want to figure out which indication we should go after, okay? Sounds like a question, you never think about it before, but there's somebody at Pfizer who needs to get up in the morning and say, we're going after this indication, right? Now, how does that person, think about how does this person make that decision, right? Well, the funny thing is they don't make it with a huge amount of data to date. And what we did at CytoReason is basically take this question, no data whatsoever, just a question. Which indication should I go after? And I know that the target is CCR6, right? Rank it across 182 different indications at low resolution, and then dig into 4 that looked like the most promising and rank them in a very kind of high-resolution manner. I'll just show you the screening stage. We ask questions like, which cells are really expressing CCR6? Which are expressing the ligand CCL20 and secreting it, which indications do we actually see differential expression of those cells, deconvolving a large amount of data, right? And with this information, we ranked 182 diseases and said, look, here's the diseases we think are the best, right? The top 4 of which, number 5, I have to say, is the one originally the drug was developed for, right? In the biotech that developed the CCR6 inhibitor. But the first 4 were not. And so what we did is we modeled the behavior, the cell composition, the behavior of CCR6 expression, and its ligand CCL20 across multiple different indications. And what you can see here on the left is a disease where we saw a good signal where the relationship where CCL20 upregulation actually affects CCR6 expression in different cells. This was UC. There were other indications that we looked at. The signal here looked best. And so we came back to Pfizer and said, look, we've looked at hundreds of different samples across many different diseases. We think UC would be the best thing to go this indication for. And I'm happy to say this was one of the considerations that ultimately caused Pfizer to make a choice to put CCR6 in clinical trials for ulcerative colitis. And this is something that's actually happening currently. Interestingly enough, when we did this originally in 2017, it took us close to 5,000 hours to do this work. Now, at CytoReason, now somebody at Pfizer can do this at 20 minutes on our system, right? And this is what I mean by breaking the data insight gap. We need to accelerate this to a point where asking serious scientific questions becomes something that's actually easy to do so that we can accelerate the process and make it more accurate. And so I'll leave you with one question, and this is something that drives me, right? How are we going to be developing drugs in 100 years from now, right? If 100 feels too much, think 20, right? 10, whatever feels good to you. The point is, don't think about today. Think about what this technology is going to allow us to do, and the point is that we need to start planning for this today. We need to start acting for it. And what I'll argue is that this is if you ask this question in this era, there's a seismic shift happening. Because how we think about biology in this next 20 or 100 years is fundamentally changing. If we think about the 19th century, this was a biologist, right, a naturalist. If you think about the 20th century, these were the biologists, right? And we're 20 years into the 21st century, and we're still feeling like this is molecular biology. But it's not, right? Obviously, we've totally changed because the data is now available at day one. And we need to be thinking about how we do basically any questions in biology these days. And what I would argue is that the next generation that I think we've started now is a hybrid between the human side and the data, so that we've modeled the processes so well of how we do drug development, how we do molecular biology research, that we've really gone to the next level in terms of using the computers. Now, to give you an analogy on this, those of you familiar with Garry Kasparov, right, the master chess player who lost to Deep Blue. So I knew for many years, Gary was a great chess player, and he lost to Deep Blue. That was basically what I knew, except I recently ran into an interesting story about Garry Kasparov, and that is, what did he do after he lost to Deep Blue? So the interesting thing is that he started a competition, and the competition had a human and a computer playing against another human and computer. Okay? And what happened was, from this were two interesting things. The first is that the duo of man and machine beat any person or any computer. Okay? And the way they did this is basically because the computer did all the tactics, computing millions of different options, and then the human basically chose the strategy of what do they want to do. Okay? The other interesting point is that Garry Kasparov is not the top, even in the top 10 players in this competition, right? The number one player in the world is a gamer, okay? And if you think about what made Garry Kasparov great, a lot of it was the tactics that he lost to Deep Blue for, right? So it makes a lot of sense. And from this came Kasparov's law. And what Kasparov's law states is that an average person with a good computational model of the process that they're trying to do is going to beat any expert who has a poor computational model. And this is where I think we're going with molecular biology research, and particularly in drug development. The processes that take to develop a drug are quite fixed. We can model them very well, given all the data that's out there. And once we've done that, we can actually make this much better and beat anything that has come before. Thank you. Yeah, Q&A. But you don't have a speaker. How do we do this?
Okay, so we're now going to open it up for discussion. I welcome questions on either of our talks, or any other thoughts you want to share. Please. Yes.
Thank you. Good morning. Thank you very much for these wonderful presentations. I have one question for you, Dr. Shai. So you mentioned the relationship between gene expression and cell types. Is this a confirmation of some biological observations?
So the way I view this is, and the context in which I mentioned it, is a technical one, in fact, right? So if you think about most assays that we do, if we're thinking about mass spec or RNA-seq, a very common way of measuring gene expression data, right? Microarray used to be. That data if you collect it, is basically you take a biopsy, doesn't matter if it's blood or tissue, and you blow up all the cells, and you measure the mRNA, okay? So what you got, and we've shown this before, is a linear relationship between the mRNA abundance that you measured and the composition of the cells that you have, right? So you're reading 20,000 genes on a sample, where the biggest variant between the different samples that you're looking on is the cell composition, right? So think about if we were measuring blood, the two of us, and you have 10 times more B cells than I do, every gene expressed in a B cell is going to look in this assay as if it's differentially expressed, as elevated in your blood versus mine, right? That, to me, is a place where we make a gap between the data insight, and in fact, we decrease the signal significantly. The way I would do this is, okay, I want to know what the cell composition of it is that you have, and I want to know what are the cells doing. Are your B cells upregulating a gene more than my B cells, right? Those two pieces of information, because of a technological gap, have been coupled together. And when they're coupled together, what happens is that an expert, a biologist, sits in front of the data. They have hundreds of thousands of data points, and they're trying to make sense of it. And in the back of their mind, they know the genes are not really telling them what they want, because there's this cells, and what you need to do is basically bring the data to think in the natural way that we as biologists and physicians think, right? So the natural way, and what we've done with algorithms, is to say, okay, this is the cell composition, this is the cell state, right? And we do this for every data type. Doesn't matter, like we bring it to the natural way that we think about the data as about.
Yeah. So can we imagine tomorrow a machine learning algorithm able to make a difference between, for example, cancer cells and cancer stem cells?
Yes, if we have enough data, I would say so. Yeah, certainly. And what
Okay.
I think cancer stem cells is still something where we don't have enough data, but I would say that would be possible. I think this is the era we're going into.
Yeah, you think that the difference will be clear using machine learning algorithms?
I think the difference will be clear, and I think the question is, what will we do with this? That should be the question we ask. Once we discover, what was the next step? I think we've been doing computational biology as a field. I think has been advancing as an art for 25 years. We've done enough art. There's enough artists here in the room. We can basically do the mental cognitive process of what is it that we do? Can we actually automate it? And I think the answer is for many, many things we can, and that would be a breakthrough for, that's what we've been trying to do in any case.
Great. I have a second question, if you allow me, regarding the dynamics of the disease. How important is this trajectory alignment do to improve your results?
Huge amount. We can pick up like tenfold difference in differential signal that we couldn't before. And we can predict, we've done this now in multiple diseases, we can predict outcome better on molecular information much better than we can based on clinical staging, especially when you go to diseases where the clinical information is poor. Vaccine responses, cancer staging, you can do way better with the molecular side.
Thank you so much.
Okay, yeah, oui.
Thanks. First, I want to say 2 amazing talks that really took us 5, 10, 20 years forward, so that was amazing. I'm trying maybe a question that is for both of you on the question of insight. So I'm trying to understand what it means, and does it mean causality? Does it mean moving from, I understand, association between A and B, to I understand that if I change A, B will change? I imagine that's a definition of an insight. And then if that's a definition of an insight, do you see a future in which we can get insights from analysis of data without proposing new experimentation? Because I thought in Eran's work, Eran touched on several points in which, hey, I see an association, and that to me is a suggestion of an experiment in which I will go and change A and see what happens. I will go and there's a strain in the microbiome that is associated with BMI that creates an hypothesis. If I add that strain to a person with high BMI, then the BMI will go down. Do we think of a future in which we use the data to create hypotheses that then are tested, or we use the data to verify those hypotheses on day one?
I think, yeah, very good question. I think, I mean, you asked, kind of, is insights always causality? So I think we'll hear a lot more on causal inference later on in this conference as well. But I think we want to use data to do useful things, okay? And I think one path, which is definitely a traditional path, is causality, right? Because ultimately we want to understand, right? We want to maybe use the data to generate insight, exactly like you said, that if I change A, I'll have an effect on B. And I think that's one path going forward. But it's not necessarily the only path, right? Because I think in some cases we'll see that data, without us even fully understanding how the machine does its work on using the data, is going to make useful predictions that just using these predictions, you'll see empirically, it actually works. It actually this algorithm is able to predict disease, and maybe some other intervention early on can help and change the trajectory of the disease, even if we don't understand fully the reason. Maybe we think we understand, but we won't fully understand it. So I think causality is definitely one approach, but I think it's not going to be the only approach. And as we accumulate more and more data, I think even just the machine learning outputs in themselves are going to generate useful actionables for us.
Maybe getting a bit more precise on that. Maybe there's a distinction between diagnostic and
Right,
treatment.
for sure.
So for diagnostic, I can see for what you're saying, that the association we maybe don't understand, but they're valid in some model, and we can use them even without an insight. For intervention, I actually wonder, like, do we actually see a world in which we look at all of your data, or all of the data that Shai showed, and we say, hey, that drug helps to do something. I don't need to verify anything anymore. There's no experiment needed. It follows from the data. Is that possible?
Yeah. I think in theory it's possible. So I understand. I think in theory it's possible. I personally think we're far away from it. But so I think it's a complex question also in terms of even if we feel confident that this is the case, you know, we have regulatory bodies, right? And will they ever? Are we close to a stage where a regulatory body will allow some drug intervention without performing a trial, you know, by the book? I think we're very far from that. But maybe as we accumulate more and more evidence from the literature and data, like the types of analyses that we're doing, there's going to be a lot of practical things that people will do, which are interventions. Maybe they don't fall under the standard, you know, regulatory book of new drugs, but interventions that people will do, which will actually be very useful. You know, I think. One example of this is diet. Okay, I think diet has an immense effect on. I think it's the cause for many diseases. I think it can be the cure. It can be preventive for not all, but for many diseases, and it's completely unregulated, right? And people follow many different. There's many different types of diets and diet regimens, and some do work for many people. So people are trying, and I think that's an area where if we accumulate properly a lot of world-based evidence data, I think we can come up with algorithms that people, you know, will be able to try, and that in many cases they will work. And as the algorithms become better, and as data accumulates more, they'll work even better and better. And those will not be with by the book standard traditional experimentation, just because historically the regulation is very different.
Thanks.
Thank you for the amazing talks. I have several questions. The first one is for Dr. Iran. Could you please tell how many time points do you currently already already have for transcriptomic data for these 10,000 healthy people?
Yeah, so for this entire cohort, we started about 4 years ago. It's now close to 11,000 people that we profiled, and about 3,000, a bit more, have come for a second time after 2 years. So we have data for 11,000 people at baseline and 3,000 more at a second time point.
So it's 2 points now.
At the moment, the majority is two points, and the second time point is being accumulated for more and more people, and some, including myself, have already come for a third visit after 4 years, but it's growing.
And you are making the full transcriptome, right?
We're doing full RNA sequencing in bulk, yeah.
Yeah, okay. And what is the age range in this healthy cohort?
So the baseline cohort is started between the age of 40 to 70, and now it's aging, but that's how it started.
And you said that you are planning to open your data set, so how are you planning to do that? How it will look like? So
it's going to be on a cloud-based platform. So we're going to open basically research rooms where you can go in, you can get access to the data, you can have all the software that you need, Python typically, but other software to analyze the data, and then gain insights, and whatever summary statistics you come up with, you can take, but you won't be able to download the individual raw data.
And train models as well.
Yeah, and train models. Yeah, basically a cloud-based environment where you can get as much compute as you want on top of access to the data that I showed.
Thank you. And another for Dr. Shai. The difference between mouse and human is very interesting, especially in aging research when we try to investigate some drugs that extended a mouse lifespan. And I'm leading the AgingNets project in Open Longevity, and there we believe that the most important thing that causes the difference in lifespans of different species is the peculiarities of topology of gene regulatory networks, so how genes are related with each other. So, you said, so could you please tell more about the model you trained to, so what, this model that dealt with mouse and human diseases, what exactly it predicted, and how exactly like training set looked like in more details, if it's possible.
So basically what we did is, this is a few years ago, right? So we worked on this for a few years to try and understand where is there enough data to try and do a machine learning kind of analysis of the cross-species gap and kind of overcome it. And we ended up deciding that the place where there's the most data that's available is in gene expression, bulk gene expression. And what we collected is we downloaded from Geo all the human disease data and kind of health, disease versus healthy control. And then this was extensive work of looking in Geo for mouse models of this disease. Okay, so if somebody published a paper that said, I'm using this mouse as a model to study disease X, we're like, okay, the only question is, can we help this person by somehow inserting a machine learning component? And so we downloaded this information and we paired human diseases with the mouse model. Okay, and so we have, if you think about 20,000 genes, and we had a couple of hundreds of such pairings of human disease, how the gene behaves, disease versus healthy, and the same in the mouse model. And what we use this as the training set to learn the relationship at a gene by gene level, how when the gene changes in the mouse, what happens in the human. We did this, it's the simplest thing we could think of. It just took a huge amount of time to do. So at that point, we're like, okay, can we do this simple? So it goes gene by gene. We've since improved this, of course. Gene by gene, and also we did pathway level, just learning this relationship. Now you give me a new mouse model, and you measure its gene expression profile. So you can rank all the genes, say the most differentially expressed gene in the mouse model compared to a healthy mouse, and the least, okay? What the algorithm takes is basically that input and the prior of everything we've learned, and it re-ranks the list. And it says, why does it re-rank it? It re-ranks it because it's seen observations that are telling it that when the gene is high in the mouse, maybe it needs to be down, it actually is downregulated in the human, right? Or, or smaller magnitude and so forth. So we're learning this relationship and then just applying it and then predicting basically what are we expecting to see in the human. We're now taking this to next level, both in kind of looking at this in the single cell level and as well as in aging. My lab actually works on immune aging, so we've been tracking how individuals change for over 20 years now from the immune side, and now we're adding in the mouse component to do this relationship. But I think it takes fundamentally to have that data in place to be able to learn those relationships. So when we did this back in 2018, I think we started working on this a few years before, there wasn't enough data available on mouse models of disease, which is something we have a lot of data for, right? For it actually to be worth that the training set would be by disease, right? So you could think, oh, well, I need to build a training set for sepsis and a training set for autoimmune diseases and cancer and so forth. Performance was actually better when we pulled all the mouse models together, both because I think of conserved regulatory networks, as you said, as well as there's just not enough data out there. I think now with single cell, there's going to be more that it becomes much more relevant to go by indication.
So you predict how the particular disease will look in human,
Yes.
the gene sets?
Yes.
Okay, thank you so very much.
Sure.
Hi, thank you for the incredible talks. I wonder if you can comment on the penetration of this kind of thinking to pharma, this very data-driven way of thinking, when it will become the major, I would say, decision-making philosophy, and what will be your best advice to younger entrepreneurs that are facing pharma?
Oh, the last one is the most difficult. I guess it would be never give up. But I'll tell you, I think, so I was at JPM 2 weeks, earlier last month, right? So this is the big conference for the pharma industry. And I basically saw something that from a side reason perspective was a great kind of nice, I would call product market fit is what they would call this. And what we saw is this. The industry is realizing the data isn't the solution, it's the problem. Okay, so in a data-rich world, the problem is how do I get insight from it, right? So if you're, so if you think about it from a business model, if you're selling data and the other side needs to get value from it, you sold it for a high price, they needs to get value for that, and you gave him a limited amount of time, not a trivial thing to do, right? Also, the industry is shifting. So if you think about, there's startups that want to be pharma companies. If you look at the old school pharma companies, they don't want to be AI companies, right? They want to be AI-enabled companies. So from a perspective of, is the industry going to build the solution? Is the pharma industry going to build the solution to solve this problem? I think the answer is, given the challenge, the size of the challenge of building, the cohorts, the analytics for this, no, they would rather buy it from the outside. Their business is to develop drugs, not to solve this problem, right? And so they can only, that means there's only so many data scientists they can hire, right? There's only so much they can do internally to do this. They'd rather partner to do this. So I think those 2 points, that the data is the problem, not the solution, and that you don't want to develop this inside, and there's only so much that you can increase inside, is actually something that I think is really putting the field of computational biology in a place where everybody recognizes that it's the way now for insight. Like this idea of, we'll just generate the data and solutions will come out. You know, I said this when I started. You know, I've been doing this for 25 years. Basically, my company is built out of frustration. And it's the frustration that, you know, we were promised that everything was going to be solved. And you look, and it isn't. Now, why isn't, right? So you need to industrialize what we do. And I think that's, at least to me, this, I suggest the industry is ready for this.
Yeah. No, I fully agree. And but I would add to that, though, that if you think of, if you look at the leaps that we made in understanding, they also came from having new types of data. The ability, technological advances that allowed us to measure things for the first time, they also drove development. So I think now we're at an era where we can finally really profile people, we can profile people and at a large scale with new technologies. And I think, yes, we will of course need to, it'll be more challenging, we'll have more data, we'll need to develop the methods to analyze it, but that new data is also going to generate new low-hanging fruits just because we've never looked at some things that for the first time we're looking at. So I think it'll be the combination of having the data, but then yes, also all the analytics for it.
I'll just add what Iran is doing, no pharma company is doing. Nobody is generating the multi-omic data that's going to allow you to slice through the, the large complexity of this, right? Just tells you, and people are interested. It tells you what they're willing to do and how they recognize it.
Yeah, thanks, Harry, for the great talk. And well, I have maybe a perspective kind of question of so what you kind of showed is basically mining lots of papers, knowledge bases to kind of summarize the data, which can potentially make decision-making easier for pharma. But I think in the end, it's technically would be summarization of the data, or you also think it's kind of also inference. If it's inference, so I just want to know, how do you see the future for this? We have this large language models and now predict stuff, but they also have a lot of problems with even the type of stuff that they generate. There are a lot of misinformation in there. I think with biology it could be even worse because let's say it's not super easy to translate from even in vitro model to in vivo or mouse to human. So how do you see the next step for this? Do you think also inference would be added here? And then if that would be the case, then how do you account for those kind of differences in hard cases?
Sure. So the knowledge piece is just one component of what I showed. Basically, what we've been building, and I think is the solution, is bringing in the data and the knowledge, right? The reason you need the knowledge is really for two things. One is to narrow the search, basically as priors, narrow the search space and kind of allow you to build confidence. That's the second piece, right? If I gave you an amazing solution, I said, this is the best target to develop a drug on. And if I'm a person who's been developing drugs for 20 years, I'm going to say, what do you know about this? And you say, nothing, but my machine learning model tells me this is an amazing target. The answer would be, take this someplace else. You need to be able to link all your new insights from data to the information that's been coming, that's been available in the literature. And so I think it's an integration piece, and the literature mostly plays the role of a prior here, as well as adding some directionality, allowing us to teach how people build hypotheses, as Rui was asking, right? Ultimately, I do believe, we're basically, machine learning is going to slice drug development 1% at a time, right? 5% AI across all drugs, 6%. And so this is how it's going to build. And the literature has a critical component of helping us to understand how we build hypotheses for this.
I think we should end right now because we're out of time, but we had a great discussion, I think. And thanks, Shai. And we'll rejoin at 11 for the next pair of talks.